Good morning. Bow your heads with me, please. Father God, we thank you so much for today, Lord. We thank you for the privilege of homeschooling. We thank you for the privilege of living in the United States of America. Please, Lord, guide our nation so that we might be a country that brings you honor and glory. Please help us now, Lord, as we do this class, that we might be awed at your amazing creation, Lord, and that you might be glorified as we look at the things you've created. And thank you so much for that privilege, Lord. And please bless all the families represented here watching this, and whatever their needs are, please provide for them. Thank you, Lord. In Jesus' name we pray. Amen. Now, the reason I'm filming this class without students, which is going to make it very difficult for me, is because of the coronavirus. And so for those of you that are watching this at another time, please be aware that we're filming this in early 2020. And so we're dealing with the coronavirus. And because of that, the CDC, Center for Disease Control, has asked everybody not to gather together in groups of 50 or more. And therefore, we can't have our live classes right now, but we do need to finish our school year. And therefore, we're going to do it this way. So I'm going to try to act like you're in the classroom with me, which is going to be difficult because it's extremely empty and quiet in here, but that's okay. I'm going to try to do it like you're here. So as we do the review, I'm going to hesitate so that you can answer at home, hopefully, that you know the answers. And so I would like you to try to participate in that way. And then if you have questions or comments that you think might be a blessing, please email them to me, and I will either email an answer out or I will address it the following week when I film class, because I still want your participation, please. And so please go ahead and email any questions that you might have or any things that you might have to add to it, because I learn a lot of the information from you guys, because you guys live in Florida and you've been around and seen a lot of these cool things. And so please don't hesitate to send me emails so that I can address those and include those. Anyway, okay, so last week, well, let's do a quick review because we didn't get to do a review last week. Last week's class, class 24, was actually a class I had filmed back in 2016. I was not aware we weren't going to have class until Friday, and therefore I didn't have time to actually film and process a whole new class before I had to have it up on Monday for the HEED students. Therefore, that class, I was blessed to have a class that covered that particular chapter on submarine meadows, but it wasn't what I had planned. So we're going to be doing a lot of extra stuff today to make up for what we missed last week, including a very quick review. So please work with me here. First off, you all know that it's one large ocean. And so the Earth's largest ocean, the name of it is the Pacific Ocean. Good. And then the ocean that has the longest mountain range that we live on is the Atlantic Ocean. Very good. And then over here, this is the Indian Ocean. How about down here? The Southern Ocean. Good. And how about the smallest ocean on planet Earth? That's the Arctic Ocean. Very good. So we name it 5 different things, but it's actually one large ocean. And then we learned about the zones in the bottom of the ocean, and we learned that if this is your sea level right here, then this area right under the sea level that's still shallow is called the continental shelf. Good. And then going down, it's called the continental slope, and then at the bottom of the continental slope, and I'll go over here for this, and it's not in this picture, it's like a pile of sediment called the continental rise. If you have mountains underneath the ocean, so submarine mountains, they're called seamounts. If they're flat across the top, they're called geos. It looks like guyot, but it's a geo. That's how you pronounce it. If it comes above sea level, then it's an island. If you have an area that is spreading, this is a rift, R-I-F-T, but this is also a rift valley, okay? And there was one other name for it. Oh, ridge. There you go. And so this is an area where the plates are moving apart. Here we have subduction going on, so this is a trench. And then this we said was the abyssal plain, this area in here. So you do need to be familiar with those zones of the ocean. And then we've worked our way from the highlands in Florida out to the edges, and now we're off the edges and we're into the water. Remember that we have mostly high energy shorelines on the east coast of Florida, therefore we have the most beaches over there. We do have some high energy shorelines on the west coast, but not as many. We have more low energy shorelines, remember it has to do with how the steepness of the land coming out of the water. If it's not steep at all, then it's a low energy shoreline. If it's got more steepness to it, it's a high energy shoreline. And so over here we have a lot of low energy shorelines. In the northern part of the state, we said there were more, you tell me, are there more marshes or mangrove swamps in the north part of the state? Good. More salt marshes in the north part of the state, more mangrove swamps in the southern part of the state. And so we identified this pioneer plant as sea oats because it actually helps to hold the beach in place. And this would be a high energy shoreline. Here where it's actually the intertidal zone because it is between the tides, between low tide and high tide. This intertidal zone at this particular beach is called the foreshore. Back here that's higher than the high tide, therefore it's very dry, is called the backshore. And then this is called sea rack. Very good. And that is the base of the food chain. And there's all sorts of weird things that can be found in there to be eaten and to look at and be careful of man o' wars. You know that. This is a salt marsh, and there are different zones in the marsh depending on the height as far as the sea level goes. This was a keystone species in the salt marsh, which is the fiddler crab, and they actually aerate the soil and turn over the top inch of the soil every year, which is amazing. They're also a food source to a lot of critters. And then this is your South Florida low energy shoreline, which are your mangrove swamps. And that particular set of mangroves, these are red mangroves. You can tell that from the prop roots that they have. And they have lenticels on the prop roots. Remember, those are like nostrils that open up at low tide when the water level's down and then close at high tide when the water level's up. And we saw that the black mangroves, which have these pneumatophores, which are stick-like roots, they too have the lenticels on them when the tide comes in, and these don't live right on the water. They usually live a little farther back. But if the tide comes in and covers them, the lenticels will close, and when low tide comes back and they're exposed again, the lenticels will open and they will breathe. And I wanted you to know what this fish is. It is a snook. That's right. So remember that one and start noticing it when you look on cars and things, t-shirts, things like that. And this was a Jewfish. It was one that we saw in the submarine meadow areas. And then that was I'm so sorry. We saw the Jewfish when we did the estuaries, the second part of the estuaries. And then this was an American crocodile, which we do find in salt water. So the Florida estuaries that we were told about in the book had three different possibilities as far as the base, what the base is made of. It can be mineral-based, which would be either loose sediments or solid limestone bottom. It can be plant-based, which means seagrass beds or algal beds, which is actually what we studied last week when we did the submarine meadows. Or it could be mollusk reefs, worm reefs, sponge reefs, and coral reefs, which are animal-based reefs, and we'll be going into that shortly. We're not quite there yet. We said that in estuaries that you have the fresh water coming out. Remember, an estuary is where the fresh water comes out into the salt water. So we have many estuaries on the west coast and about 4 major estuaries on the east coast. And remember, estuaries are places where the fresh water rides on top of the salt water, and then it kind of does some mixing and turns into brackish. But remember, in the estuaries, the life in the estuaries have to deal with constant changes of the temperature, of the water level as far as how high or low the water is. And that's got to do with not only the tides, but whether you have a lot of rain inland or droughts inland, etc. So that can affect. We had ever-changing salinity, which is the salt content because of the fresh water coming out and into the salt water. And remember, when it mixes, it's brackish. And then we had differences in the turbidity. And so that had to do with how much sediments were in the actual water. So things that live in the estuary have these different changes to deal with on a constant basis, so it can make life pretty difficult in the estuary. Salinity changes, water level changes, temperature changes, and turbidity changes. And so they make life kind of hard for them. And then we looked at zooplankton and phytoplankton. Remember, the zooplankton are little tiny animals. The phytoplankton are little tiny plants. And if those little tiny plants have glass houses, we call them diatoms. And then we looked at nudibranches, and we saw that they came in all sorts of cool colors. And these are really how they look, but I had no idea they were so small. And the reason is, remember I told you that I caught one? I found out from the reading I had a sea hare. It wasn't a nudibranch. Nudibranches are smaller. Sea hares are classified with them, with sea slugs in general, but they're bigger. They can get up to 5 pounds, and that was what I held. I got a picture of it for you, so I'm going to show it to you today. Anyway, so those are the nudibranches, and within the nudibranches, there are the aeolids, which you read about on plate 100, that are actually able to eat sea anemones and things like that, and they're able to process the poison dart cells, which are called nematocysts, or cnidocytes. They are able to process them in such a way that they don't get digested. They actually put them out into their cnidosacs, into these tentacle-like things, and then they can use them as if they're their own. So they've recycled the poison dart cells from the anemones and corals and any other cnidarian that they might eat, and we're going to study those soon. And so I thought that was pretty cool, and that was on plate 100 that you read about. And then a couple weeks ago, we started into echinoderms. We said that meant spiny skin. And we did a starfish dissection. We were introduced, for those of you that hadn't seen them live before, brittle stars. Most of you have seen sea urchins, sand dollars, sea cucumbers. We had a opportunity to look at those. Once again, we dissected the starfish, so we became aware that it has a boral side. The oral side is where its mouth is. The aboral side is the other side. It has a madreporite on the aboral side, where the water is pulled into this water vascular system that they have in their rays, and that they're able to use those 2 feet to not only move around, but to grab their dinner. And remember, when they grab their dinner, I have a picture. When they grab their dinner and open up the clam or the shellfish, then they'll actually invert their stomach into the prey and digest the food and then pull their stomach back into their body. So pretty weird. Anyway, there's some really interesting stuff in the oceans. And then this was a sea urchin, and you could actually look here and see the tube feet on the sea urchin. And we saw that they have a very interesting mouth, and the apparatus that makes up the mouth is called Aristotle's lantern, and there's 5 teeth involved, and they are grazers, actually. And that if you cut one open, you can see the 5, the radial symmetry, these 5 points, like you would on a sea star, and that's why they tend to be classified, because they do have some similarities. And then we looked at sea cucumbers, and we saw that sea cucumbers actually eviscerate themselves, or extrude, put out, throw up their innards to defend themselves from something that might be trying to eat them. And then these tentacle type things that they're throwing up actually have toxins in them and get very sticky like tape, so that whoever's trying to attack them themselves might die. And so that's their form, sea cucumber's form of defense. And then this was a larva that's going to turn into a brittle star. So you tell me, is it a meroplankton or is it holoplankton? Okay, some of you have already said it's meroplankton, because holoplankton stays plankton its whole life. Meroplankton changes into something else, and this is going to change into a brittle star, so that is a form of meroplankton. And then that is going to turn, believe it or not, into a crab, so that too is meroplankton, because that was a crabzoia, and then that crabzoia will turn into a megalops and eventually start looking a little bit more like a crab. You know which type of crab that is. That's a blue crab, that's right. And is it a girl or a boy? Look at the red on her claws. That's a girl, isn't it? And then this was a mero larva of a sunfish, which turns into this monstrous fish. And it's hard to believe, because this is like almost microscopic, it's so small. So just incredible how these things can grow. And then that was 2 weeks ago, we looked at how the sea anemone can actually protect itself from the starfish. And that's a sea urchin. I'm sorry, the sea urchin can protect itself from the starfish. And we saw the really cool video on how the sea anemone actually swam away from the starfish because it wanted to save its life, which was really cool. And then we saw how the scallop, the bivalve, 2 shells, was able to get away from the starfish as it tried to eat it. So we saw some defense mechanisms there. And then we learned that gastropods, that mollusks are soft-bodied creatures. We learned that mollusks, it's called mollusca. The phylum is mollusca, or mollusks. You can just put a KS there for mollusks. They're soft-bodied. They usually have a some kind of a hard shell, even if it's just their beak in the case of the squid or the octopus. And so we said that if mollusks had 1 shell, they're gastropods, because they gastro meaning stomach and pod meaning foot, and they walk on their foot. So gastropods, like snails and conchs. And then there were bivalves, those are univalves. Bivalves have 2 shells, the valves are the shells, so that'd be more like clams and oysters and scallops and things like that. And then when we looked at the reefs, we learned that there was, well, do you guys remember what that's made by? What kind of creature makes that? And it's not coral. That's a worm reef rock. And I've seen these before, and they're made by worms. And I didn't realize that worms built rocks and reefs. But they secrete a protein that they can glue the sand together and make these, and then it turns into a worm reef. So there are worm reefs around Florida too. And then we looked at the seagrass beds last week, and we saw that the seagrass is actually a regular flowering plant. So they actually have flower, put pollen into the water, and they have to be pollinated and release their seeds, etc. We saw that there are all sorts of types of Meroplankton and holoplankton. All of these little plankton actually serve the same purpose as bees and butterflies and birds and bugs. They're pollinators. They can actually help to move the pollen to the other flowers so that it can be pollinated and seeds can form and the plant can continue to propagate. So they're important. And then also they can go through asexual reproduction, which is like a form of cloning because they have these rhizoids, and then they will grow a whole new plant off of these rhizome runner type things, these not rhizoids, rhizomes. And so they're able to grow a new plant off of those. And then we looked at 4 specific types of seagrasses. The shoal weed, which is a thinner leaf, grows closest to the shore around the mouths of rivers. They can tolerate low salinity, so a lot of fresh water, and they can be exposed to air for a certain amount of time without dying. They do help the sediments to hold in place with their roots, and then they help to build land. This is turtle grass. This is that wider grass. And I don't know if you can tell, but each leaf has a lot of things growing on it. So each leaf is its own little ecosystem, honestly. And turtle grass grows below the low tide mark, so it can't be exposed to air for any length of time and still survive. It needs to be underwater. And they need a lot of sunlight, so they only grow up to about 10 feet deep, because when you get deeper than that, you have less light. They tolerate low salinity, so they can handle that influx of fresh water. So you'll find them near the coast in a lot of places, but they have to be underwater because the exposure to air will destroy them. And this should say that their roots stabilize the sediments. I messed up here. So anyway, so their roots do stabilize the sediments. And then there's manatee grass, which is a little deeper still. And the manatee grass grows from about 10 feet deep, which is where the turtle grass stops growing, to about 30 feet deep. And they die if exposed to air. They have to stay underwater. And then this type was Caribbean grass. And the Caribbean grass could grow all the way to 100 feet deep because it can handle low light. And sometimes it will be the understory plant. So in other words, it will grow underneath the turtle grass and the manatee grass. So you will see this one scattered in between. All of the sea grasses lower the turbidity by preventing the bottom from being stirred up. They provide energy, an energy source for herbivores. In other words, you have the sea turtles and the manatees and other animals come in and grazing on this. And they all have rhizomes so that new shoots can form and grow from each node. So that is a form of asexual reproduction that they can go through. And then we saw last week that there were algae that are macroscopic algae, which Floridians call seaweed. This particular one is Sargasso seaweed, and that's what it looks like as it's growing. And I've seen this in places in the Keys off of Isla Morada, right along, not too far off the shore. As you're going south, you'll see these coming up to the surface and then just kind of laying over in the water. And a lot of sea turtles, usually about dinner plate sized sea turtles, that'll be around in there grazing on them and stuff. So it's pretty cool. And you guys see these on the beach on the East Coast. This is the type of seaweed that you see on the beach, and they've got the little pneumatophores that you can pop because those are the little floats. So as a kid, I used to like to walk along and pop them. I think most kids do. Anyway, it's like popping bubble paper. Anyway, and then we talked about how there are critters that are perfectly camoed. They're designed by God, camoed, so that they can hide in these. And once again, an evolutionist would say it adapted to this, but I would like any scientist to show me how the DNA changed to tell it how to look like that seaweed. No, it had to already look like that seaweed, and then it hid in it because it was a safe place for it. So I agree that it's adapted to its environment, but I believe it's adapted because of its DNA, had the genetic information in it for it to have those adaptations for it to live there. So I just wanted you to understand that. And then that would be an example of another macroalgae that looks like red algae to me. And we do have that here also. And then this was a polychaete flatworm, and he's in the seagrass beds. There are polychaetes in there. There are squids in there, and I don't know why, but that really looks more like a cuttlefish to me than a squid. And I think it is a cuttlefish, but it's supposed to be a squid. We don't have cuttlefish here in Florida as far as I know. And then we do have seahorses, which are so cool. And then we have a lot of rays, and there is one of the things we read about this week, and I didn't even catch it that I got this in this picture. This is a ray, and look at the barjack that's with him. That's that symbiotic relationship we read about. We're going to cover it in a little bit. We're not covering it just yet. But that was the seagrass beds. And the bango shark was one of the things in the seagrass beds. So see, apparently I had a few things that didn't live here in Florida in these pictures. And then there's your scallop and his 200 blue shiny eyes, which I find unsettling. And there's your sea turtle, which loves to eat sea grasses, turtle grass specifically, and we'll come back to that when we do turtles. Okay, let's see. Oh, and sometimes you'll find the gastropods in the seagrass beds. That's where I actually see conchs. That's what that picture was. If you're snorkeling, here is the conch. Can you see him? So as you're snorkeling along and looking in the seagrass beds, you have to really look for the conch. And then if you get down near him, don't flip him over necessarily because it can cause him problems. But if you get down near him, you'll see his two little eyes coming out the side, which is very cool. Anyway, and then we read about the life cycle of the, what I call Key West pink shrimp. And so we saw that they started as an egg and that they went through that huge journey that they do between the coast of Florida and the dry Tortugas and back. And we saw that only 1 out of every 10,000 actually makes it through the life cycle. So there has to be a huge number of shrimp. So we did that last week, and we will talk about turtles and manatees. Actually, turtles we'll do more. And then there were several different types of wading birds that we see that live on a lot of the produce from these submarine meadows. Okay, so that brings us up to some of the stuff we didn't do last week and some of the plates that we were supposed to read last week. I know one of them was plate 42. So I'm going to turn to plate 42. And plate 42 you read 2 weeks ago. And that was on sea squirts and larvaceans. And these are sea squirts. And sea squirts actually start as a tadpole-looking larva, and then they turn into this little thing. These are multiple sea squirts, so they're called, if I remember correctly, they're called social sea squirts because there's multiple ones. And see, they have an opening here, and they squirt the water out through there. So they're filter feeders, and they can be very colorful, and once again, they can live in groups or they can live individually, but those are tunicates because they have like a leathery tunic. And so they're called tunicates. And you'll learn a little more about those when you get into biology. And then this was, these are the compound sea squirts. Okay, so these are social sea squirts. So when you see multiples together, they're called social sea squirts. This is an individual sea squirt. And then these are compound sea squirts. So they actually, if you look at the picture on plate 42, they're kind of in the middle of the picture. You see the compound sea squirt, and they share certain parts with one another. So anyway, those are sea squirts, and they can be pretty interesting looking. And then if you go to the next one you were supposed to read, which is 32, shellless gastropods, on plate 32, shellless gastropods. Here, you have the nudibranch, which we've really already talked about, and the aeolids, which we really already talked about. But there's the sea hare on the bottom right of the page. This is a sea hare. That is what I saw, and I want to give you an idea of the size. See, so I actually just reached down in the water. These parts were going, and so I just reached down under them and picked him up. And he was about the length of my hand, so it's very close to that. He might not be quite as big as that one, but it's called a sea hare because he looks a little like a bunny rabbit in his face, doesn't he? And here's his little arms that he swims with like this. Okay, so that's a sea hare. And so those would be considered a shellless gastropod, which was covered on your plate 32. So I found that to be interesting. And then, let's see what our next plate is. Okay, we already talked about the nudibranches and that they came in all these different colors. And we already talked about that they're small and that the aeolids actually are able to put the nematocysts, which are the poison dart cells, into their nido sacs and reuse them. And then on page 79, on the very last paragraph, it said that these creatures are what's called hermaphroditic. Hermaphroditic. Now, a hermaphroditic animal or hermaphrodite actually has both male and female organs. And some of you are thinking, what? Well, earthworms have both male and female organs. They can't reproduce with themselves. They have to exchange with another earthworm, and then they have the information that they need genetically to make an egg to have a baby. And that's how these guys are. They too, like an earthworm, are considered a form of worm-type creature. These are actually gastropods, though, whereas worms are classified separately. But the point is, we see that these two have to exchange sperm with each other for the eggs to be fertilized, and then they can make the eggs with the baby in them and release them. And so you'll see in the ocean, there's a lot of different examples of hermaphroditic individuals. There are fish that can change from male to female or female to male as need be. And some of the students were kind of like, what? When I mentioned this once before. But if you think about in the ocean how hard it might be to find a mate, that's why God made it so that these animals could flip-flop from one to the other. So, and I don't want to get into that in the actual anatomy of it because it's too difficult for our class, but I wanted you to be aware of it because once again, when you get to biology, or if you've already had biology, then you've already studied how earthworms are hermaphroditic too. So we see that here also, which I found kind of interesting. Anyway, so that was on plate 79 at the very bottom of the plate. And then review-wise, we already saw that the polychaetes, and this is the polychaetes, can have a proboscis that can have some pretty mean pieces on the proboscis so that they can grab their prey. So for a little worm, they can be kind of frightening if you magnify them at all. And so that was our polychaete. And I have to apologize. I was going to do your observation on your polychaete with you today so that you could write up that lab. But I think I already took it to Boca Raton knowing I was going to use them for the lab, and therefore I don't have them here in Naples, so I can't film that lab for you, and I do apologize. But we're going to do the skate lab together today because I have that here. So I just wanted you to be aware of it. So I apologize that you could always draw the polychaete from a picture on the internet if you would like. But that's the best I can offer you at this point in time, and I'm sorry. Remember, the polychaetes do have tentacles that they filter feed with, and so these are called the radioles, which are some pretty nice looking worms as far as their tentacles and how they help them. Okay, then one of the plates you were supposed to read, plate 77, was about the reproduction in the polychaetes. And the part that I wanted you to see is just that very first paragraph, because in that first paragraph, it tells us, it says, experiments done with polychaetes and sea stars suggest that invertebrates, like these animals, possess a mechanism that allows them to track changes in length of day and synchronize their annual reproduction cycle accordingly. They spawn, their spawning time comes closer. Some combination of the monthly moon influence changed in tidal range along with changes in the amount of moonlight is thought to be the external cue that synchronizes the spawning of the invertebrate population to the moon phases. First off, it says moon phases, so let's make sure you understand what moon phases are. Remember, well, this is the length of day. Remember that the days are longer in the summer and shorter in the winter, and it's got to do with the tilt of the Earth and where we are in proximity to the sun. And we're in the northern hemisphere, so we get longer days in the summer, June, and shorter days in the winter, which would be around December. So the length of days will cue them as to whether they should spawn or not. And the other thing that they're cued on is phases of the moon. And we'll see this again with the cnidarians, specifically with corals. Okay, and sponges, from what I understand, the spawning has to do. So see, you got a whole bunch of invertebrates, just like it mentioned, that actually depend on the length of day and on the phases of the moon. The phases of the moon being, if it's a full moon, you guys are all familiar with that. And that is when the sun is able to reflect its whole surface of the moon, and that's why it looks like a full moon to us. As it starts to move away or go around, then you see shadows start to form on the moon, and it gets to the third quarter, and then it gets to the waning crescent, which is actually what I saw this morning. And then it's between, the moon is between us and the sun, therefore you can't see it. Now, no light is reflecting on it, that's the new moon. And then as it starts to move again, you start to see the crescent form the first quarter, and then it eventually goes back to the full moon. Now remember that when the moon is lined up with the sun, we get a spring tide. So we get spring tide on the new moon and on the full moon. Whereas we get neap, remember spring tide, higher high tides, lower low tides. And then the neap tide is on when it's at a right angle, so that would be on the first quarter or the third quarter, because the gravitational force isn't linking up and therefore it's not pulling as hard. So the neap tide is where you have less, so a lower high tide and a higher low tide, you have less of a fluctuation or less of a tidal range. And so remember these, because actually these creatures, depending on the length of day and depending on the phase of the moon, that cues them to know whether they should spawn or not. So that's pretty cool when you think about it. Anyway, so that's why I wanted you to read that part. Then we get to today's work, finally, right? Okay, so turn to plate 63, please. And in today's work, we're talking about the coloration in fish. Advertisers. So we got some pretty doggone colorful fish out there in the oceans, don't we? And so we see that advertisers, and I don't know if you looked at all the plates. We're only doing a couple of the coloration plates. There's the advertisers, the cryptic ones, the changing colors, the advertisers. I think that's, we didn't even read the advertisers yet, the other advertisers. Because those are invertebrates, and now we're dealing with fish. Anyway, so these are some of the very colorful fish. This is, I guess he would be a fish. I was looking at the eel here. And so let's go ahead and look at some of the ones it mentioned. First off, it mentioned the advertiser, the orange Garibaldi of Southern California, and that is an orange Garibaldi of Southern California. Apparently, this is the California state fish, which I've never seen one of these. But it acts just like some things we do have here in Florida. We have the Sergeant Major fish, and we have the damsel fish. All three of these fish, so I'm just going to go back and show them to you. All three of these fish use their bright colors to establish their territory. They let everybody else know this is my rock, or this is my grass, or this is my home, and you stay away. And so they're advertising their territory. And it displays, it's a territorial display. Another fish that it mentioned here was the lionfish. And that is the lionfish. And they are no longer just in the Indian Ocean and near Australia. The story that I was told was when Hurricane Andrew came through in 1994, one of the houses that it destroyed in Key Biscayne had a fish tank with some lionfish in it, which escaped because of the flooding. And that's what I was told, that's where our problem here in Florida comes from. But we have an invasive species problem with the lionfish. They have no natural enemies here, and they are very prolific, which means they're very happy here and they breed very fast. And they are eating up, because they have no natural enemies here, they're eating up a lot of the native fish. So they have lionfish derbies now, and they try to get people to catch the lionfish, and they're allowed to sell it because we're trying to get rid of the lionfish. And it's supposed to be a very tasty, white, light-colored fish as far as the food. But we're told here that they are venomous, which means they have venom like a rattlesnake does. And so you never want to try to touch one because they're venomous in their spines. The people that are spearfishing them actually wear special gloves and take a pair of wire cutters with them, from what I understand, and clip off the venomous spine when they catch them. So anyway, you got to be careful with that. So in this instance, the color advertises that they're venomous and that they're dangerous. And that's true in those weird little frogs down in South America and stuff like that. So frequently color will warn of being dangerous or venomous. And then this was interesting, and I was unaware of this, that the angelfish, believe it or not, that's a young version of what this is. This is the adult version, and this is the juvenile version. So this fish starts looking like this, and then he grows into this adult, which I had no idea that angelfish made those kind of changes. And it actually told us here that the juvenile, let's see, it allows, okay, the color differences allow them to have quick recognition as to whether it's a juvenile or whether it's an adult. And then the book said that the distinctive coloration of the adults helps to keep bonded pairs together. This is a Florida, because this last one I think is a California angelfish, but they might be here and I just don't know. So please, I'm not sure. I'm just not aware of this particular fish. But these queen angels I see all the time, they're gorgeous. And I see them on the reef. Get this, watch. That's a juvenile queen angel, and that's an adult. I had no idea that this changed into that. So now when I see these, I'll know it's just a baby version of the other one. Anyway, so the color can actually signify whether it's a juvenile or an adult, which I was surprised at. And the last of the advertisers that you have here is the copper band butterfly fish. And the spot at the back of his body functions to be a false eye. So that if another fish is trying to eat him, he'll dive for this direction thinking that's his eye, and then the butterfly fish swims off in this direction. So all the predator fish gets is a mouthful of either back fin or water, while this guy's scooting off in the other direction. And there are a lot of land animals that have the eye type camouflage. Like bobcats have little, what looks like eyes on the backs of their ears, so that if you're seeing the back of them, it looks like he's looking at you. There are other animals that have this. So we see this kind of coloration in fish also. So that was plate 63 with the advertisers. Then we get to plate 64, and this is one of my favorites actually. This is called countershading, and I learned about this the first time I taught about marine animals, and I was really kind of, wow, that's cool. What it is this. There's a lot of animals with countershading. And what that means is they're dark over the top and they're light underneath. Now, this is very logical if you think about it. If I'm looking down from the boat and I'm looking down, if I see this fish go by, I'm not going to see him because this dark blends in with the dark water underneath him. So he disappears. Let's say I'm scuba diving and I'm underneath this fish looking up at him. When I look up at him, all I see is the white or the silver blending in with the sky behind it. So countershading works really well for water critters because when you look down, they disappear with the water, and when you look up from underneath them, they disappear with the sky above them. And so that's what countershading is called. You'll see it in penguins and in all sorts of fish. Whereas a grouper was the example the book gave of non-countershaded. Notice it's the same throughout. Well, groupers don't swim usually in just open water like the tuna and some of these fish with the countershading. So these are non-countershaded, and notice that he looks basically the same all over his body. This is a billfish. And notice it is countershaded, and it's actually got stripes across the top, and it's darker with the lighter color underneath. This is a wahoo, And the wahoo has the stripes over the top, the darker stripes over the top, and then the lighter color from underneath. And I just wanted you to see, because when I first saw this, I thought it looked something like a barracuda, but I know it wasn't. And I looked it up, and it's a wahoo. There's your barracuda to compare. You notice his face is much scarier in the barracuda than it is in the wahoo. And he does have some slight stripes, but not as much. So they look kind of similar, but not the same. Much scarier mouth on the barracuda. Just wanted to point that out. And then on plate 64, it also talks about the disruptive coloring of the clownfish, or the anemone fish is another name for it, which I did not know. And so this would be disruptive coloration, because you notice that the colors actually are changed so that he can fit on a coral reef. And there are other creatures that have these kind of stripes and stuff, so that on the coral reef, where there's so much color anyway, they can just blend in. So that's disruptive coloration. And then the last example that it had there, in the book, called it aggressive mimicry. Believe it or not, that's a fish. That's a stonefish. And he looks like the bottom, doesn't he? And the problem is he's venomous. Thank God they don't live here. They live over in California. But if you are in the Pacific coral reefs, if you step on him, he's got a venomous spine and it can kill you. So this isn't good. Good reason to wear beach shoes when you're in the water. But anyway, so that would be, the book called it aggressive mimicry. And then we go to the next plate, which you were supposed to read, and that's plate 65. And on plate 65, it talked about color changes in fish, and there were two different types of cells that they have that can help them with these color changes. One thing they have is iridocytes. Now, iridocytes, you can think of them as mirror cells, okay? Because in the iridocytes, they actually have guanine, which, here it is, guanine, which actually acts like a crystal to act like a mirror. And so the iridocytes give the fish either the white or the silvery color, maybe a silvery blue or a silvery green color, because they're reflecting back the light through the little mirrors. The other type of color cell, that's showing you how the iridocytes will reflect back the light, and this gives great details as to exactly how these things work. But the point is that they reflect back those light, and it shows you on the squid and how that helps to get the silver or the white colors. The other way that fish can change their color is through chromatophores. And the chromatophores, the book told us, actually are cells that are pigmented cells. And here, I've got another picture here. These are actually chromatophores. So this is showing you what's blown up here, and this is even a closer view, okay? And these chromatophores can either keep their, they can be contracted so the pigment can stay in one small spot, or it can be expanded where the pigment expands out and then the thing looks like it's got more color. And this is actually how a lot of fish, like the octopus, is able to actually, like a chameleon, reflect what it's going by, and it looks just like the background as it swims across. We're going to study them soon. And so it's these chromatophores that they can change dramatically and very quickly. So there are patches of these pigments in their skin that can actually open up and show more pigment, or contract and show less pigment. So they have iridocytes and chromatophores as to how they can make their different colors. Then we go over to plate 91. And we started to read about some of the different forms of symbiosis that we see in the ocean. And this was your invertebrate farmers. Now, this particular picture, once again, this is on plate 31. And this is Alicia, which is a type of sea slug. And so he looks a lot like the sea hare, doesn't he? He's a sea slug. He eats algae, and then the algae, he actually puts it into his tissue, and that's why he looks green. And then he's able to let the algae photosynthesize, and he benefits from the algae, not only by eating it, but actually by allowing it to live in his tissues. And so he benefits from the food that the algae produces in the process of photosynthesizing. Now, it's interesting, that's not the only creature in the ocean that does that. The giant green sea anemone, and I got a picture with a sea star in there for scale, so you can kind of get an idea. Most anemones are small, and these guys are apparently pretty good size. They actually are green because of the algae that they eat, zooxanthellae, and this is how you say it, zooxanthellae. And zooxanthellae is also the algae that you find in coral polyps in the Florida Keys and in the coral reefs that we have around here. So zooxanthellae, is actually the algae that gives coral its pretty colors. And so we see it here in the sea anemone. We also read about it in the giant clam. That is zooxanthellae. You're seeing the top of the clam. So the clam actually attaches itself into the coral reef. I'm sorry, I don't have another picture. You see it here, though, in your plate. And this is the lips of the giant clam, and you see all the zooxanthellae. All this color is actually from the zooxanthellae, which are the algae that live in the tissues of the clam and of the sea anemone. And then they photosynthesize like any plant type of creature would. They photosynthesize, because plants aren't creatures, they're organisms. And so it'll photosynthesize, and it makes the sugars and all that. And then the algae benefit from the waste products in the cells of the animal that they live in. And they use those waste products in the photosynthetic process and turn it into food, sugars and things like that. And then the organism they live in benefit from those sugars. So it works out very well. This is actually a picture, a microscopic picture of the zooxanthellae, and they can come in multiple different colors. And then this shows you the zooxanthellae in a coral polyp. So this is a polyp of a coral, and you can actually see in the tissues the zooxanthellae that lives there. And this, in this particular plate, it talked about how that sea slug actually benefited from the algae it eats. The giant green sea anemone does, and then the corals, and it mentioned specifically elkhorn coral and brain coral, and I just wanted you to be able to see what they look like. So that's elkhorn coral. We have both of these here in Florida, and this is brain coral. And it talked about how the polyps have to be bigger on the brain coral than the elkhorn coral, and therefore in the larger polyps they had to be more efficient at trapping the zooxanthellae. So anyway, we will come back to that when we get to corals, and that will be soon. And then page 92 was a form of mutualism also, and all these have been mutualistic symbiotic relationships at this point where everybody helps everybody else. Because the zooxanthellae gets a home, but the coral or the clam or whatever it is gets the benefit of the food from the photosynthesis, so both organisms benefit, therefore it's mutualism. Cleaning stations. Now, you know, when I was your age, I'd never heard of a cleaning station. Watched Jacques Cousteau, read books, never heard anything about it. So I'm assuming that they didn't find out about these things until fairly recently. Recently for me, not for you. And so these are cool. They've even made a movie, I think it's called Car Wash, and it's about a cleaning station on a reef. So I haven't seen that one, but I'm gonna have to. Anyway, so cleaning stations are on reefs where there are small fish and shrimp that are waiting to clean the larger fish as they come in. Now, why do they need cleaning? Because they have parasites. You know, and you actually did some reading on parasites. Here, let me just show you real quickly. Let's get up to the parasites. Okay, those are actually not parasites. You read about that there are polychaetes that live in the umbilical groove of certain starfish, and they don't hurt the starfish, but when the starfish is finished eating and starting to pull its stomach in, these polychaetes actually feed off of all the scavenged what's left, and they benefit that way. So this is commensalism because they don't help each other, but the polychaetes don't hurt the starfish. And then there are ectoparasites, which are parasites that attack the outside of marine life, and that's just a picture showing you, and this is for scale to show you the size of these ectoparasites, parasites on the outside of the body, and your book showed you that. And then these are endoparasites, which are parasites that attack the inside of a fish or marine organism, and that can be an issue also. And so cleaning stations help some of these organisms to get some of these ectoparasites off. So this is showing you a manta ray, and he's come in and he's opened himself up, including his gill slits, so that these little fish can get in there and pick off these little parasites. So that's a very important service to the manta ray and these other fish that benefit from it. This is showing you a grouper coming in and opening his mouth, and he opens up his opercula, which is plural for operculum. The operculum is that covering over his gills. So they open this up so that the little animal can get in there and clean his gills. You can watch some of these on videos on YouTube, and they're so funny because I saw one of these where the little shrimp goes in, and then he gets one place and it makes the grouper start to almost gag, which kind of struck me as funny. So these guys can be pretty invasive, but they're trying to help. And then this shows you read about the blue wrasse. And the blue wrasse was actually this little fish. He's about 3 inches long, and he goes in and he cleans out in the mouth of these. And then it told us that there was a false cleaner blemmy that looks very similar if you look at it. I don't think I got that one. That's just another cleaning station with larger fish. But the blemmy actually isn't a cleaner fish, but he looks enough like this that sometimes he will actually attack and bite healthy tissue out of a fish that's come to a cleaning station because he fools him, so that's not nice. Anyway, and even the turtles will benefit from the cleaning station. The manatee, I've seen manatee pictures of the cleaning station. So a lot of these different critters will actually benefit from these cleaning stations. Okay, then we get to plate 93, so turn the page. And on plate 93, you have mutualism. And the specific mutualism that it is going into is clownfish. Now, I got this picture because this was in the actual ocean. You notice they're not as bright as they look in fish tanks. But this is your clownfish, and that is the sea anemone that it lives amongst. Now, what was really interesting on this page was it talked about how the clownfish actually acclimates itself. So it gets used to the sea anemone by rubbing up against its tentacles, and it says that it will get stung a couple times, and then it'll back away, and then a few minutes later it'll come back and it'll do it again until eventually, apparently, it stops getting stung. And they've got some different possibilities of why that might be. The one that I've heard and that's most popular is it somehow removes a certain amino acid, which is the building blocks to a protein, in the slime layer of the clownfish, so that, and that's the chemical trigger for the sea anemone, and so that's why it stops getting stung. But they say that's one of the 3 possibilities. So we're not exactly sure why. And I didn't know they had to acclimate themselves. Like if they go to a new sea anemone, it says that they'll start acclimating themselves to that sea anemone. So they go through an actual process to live in the particular sea anemone and not get stung. Now, it is a mutualistic relationship because the sea anemone provides safety for the clownfish, but the clownfish acts as bait, which draws in food for the sea anemone. So both parties benefit. And it's interesting how many different animals actually use sea anemones as a partner in living in the ocean. The other one that I'm thinking of is the hermit crab, and this is plate 94. So if you turn the page, once again, we see here that hermit crabs can actually place sea anemones on their shell because it benefits the crab, because when the sea anemone eats anything that falls off, it can actually eat so scavenging parts, but also it provides it some protection because the sea anemone will protect the crab from certain other fish and stuff. And then the book told us about how the sea anemone, there's a particular sea anemone that actually secretes like a false shell for the hermit crab, and that's pictured in your book on plate 94, and it builds a false shell for it. And I've heard of cases where the sea anemone ends up just being the shell for the hermit crab. He ends up with this mass of sea anemones on its back, and the shell just disappears and it just hangs out in the middle of all the sea anemones. So hermit crabs have a very interesting relationship with sea anemones, and they help one another out. And then another one that was on plate 94 was the southern stingray and the barjack. And that shows you the southern stingray and the barjack. And what happens is the stingray actually eats crustaceans, which are things like crayfish and shrimp and things like that. And he crunches down on stuff like that that's in the sand. And so the jack, as he's moving the sand so that he can find what he wants to eat, the jack ends up eating everything that comes up that the ray doesn't want. And so that's commensalism because the jack benefits, but it doesn't really do anything for the ray. So that's commensalism. And then I already told you about this, that this was commensalism with the polychaetes in the umbilical grooves of the starfish. Okay, and we talked about those a little bit. So there was a picture of polychaetes on the reef. And let's see, then we get to the last portion of what you did on your reading, which started, let's see, started on 50, plate 50. So on plate 50, we are introduced to what are in this book called Elasmobranch Fish. And in what we did before, we said that bony fish were osteichthyes. Osteichthyes, I hope I spelled that right. And these type of fish are called chondrichthyes. You still got the ichthyes, which is fish. Okay, this was bony fish, and this is cartilaginous fish, chondrichthyes. And so these are all chondrichthyes because they have cartilage, skull, and backbone rather than bone, skull, and backbone. Remember, bony fish don't have to have their whole skeleton in hard bone. Only part that has to be hard is the skull and the backbone. Chondrichthyes, even the skull and the backbone, are cartilage. Remember cartilage? It gives a little because it doesn't have the calcium carbonate salts in it that the bone does. Okay? So the three things that you usually think of in Elasmobranch or chondrichthyes are sharks, which we are not doing today. Don't get too excited. We're going to do sharks, I promise. Sharks, well, at least that's the plan. Rays and skates. Now, rays and skates both are kind of similar in that they're the ones that look like what you and I think of as a stingray, okay? The difference between a ray and a skate is that a stingray has a longer, skinnier tail, and a skate has a shorter, fleshier tail, okay? And they can even be shorter than this. I've seen them that had little short tails like that, but they were still a ray. Okay, so we're going to focus in specifically on the rays and the skates today. And we are told in the two paragraphs on the right-hand side of plate 50 where the writing is that some of these skates can get up to 10 feet long, and they do have countershading where they're dark on the top and light on the bottom, which you can see the dark on the top there. And then once again, well, I'll tell you what. Let's go over to plate 52 because that's where it gets more into the skates. This is your southern stingray, which was on plate 50, which we just were. And that's the one at the bottom of the plate. And you can just see here his spine. Can you see it right here? It's in the front of the tail, and that's his spine. And you read that there were denticles, which are the teeth. That's what we call the, well, actually the denticles are the teeth that are on the skin of the shark. But we're told here that they also can do other things in these animals. So that's your southern stingray. And this is the underside. So that's on page 50. You see the underside. And here on that underside, you see the, yep, I thought so. I wanted to make sure. You see the gill slits and its mouth. Okay, but you see the gill slits right here? All right, now you won't see that, and these are claspers right here, which are used for breeding purposes in the male. That shows you, if you go over to plate 52, that shows you the Atlantic Manta Ray. These guys can get, well, let's see, what does the book say? The book says that the manta ray can get 20 feet across and weigh up to 3,000 pounds. I personally have seen a manta ray that was over 20 feet across because we were in a 24-foot boat, and it came up, and then it went splash like that, and it was gorgeous to watch it. It was kind of scary because it was so huge. Obviously countershaded, dark over the top, light underneath. The book talked about how these are cephalic. Cephalic always is referring to your head. If you have encephalitis, you have inflammation in your head. So these are the cephalic fins because they're on his head, and they actually, these are filter feeders, so they actually help the water to go in so that they can filter out the food that they need. I got to just tell you this because real quickly, when I was a teenager and we went to Bimini one weekend, and we would spend the night at Honeymoon Harbor, which was a little island south of Bimini. There were no people, no stores, nothing, no power, nothing on that island, just a rock and a lot of birds. But anyway, so in the evening we would hook the boat up and we would just hang out, you know, after we ate and just talk or stand in the water. And I'm standing in the water next to the boat, you know, and it's probably 3 feet of water, no big deal. And I see something big and black, like right there. So I grab a mask from the boat and just stuff it on my face and just kind of got under the water to see what it was. And it was one of these, and he had to be 6 feet across on his wingspan, and he wasn't more than a couple feet from me. So I kind of almost drank a lot of seawater, like, blah, 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 you know, freaked out a little, because when I got down, I saw this face looking at me, and he was just looking at me like, what are you doing, you know? And I shouldn't have been frightened, but I was, because I didn't know what it was, and it was pretty big, and it was very close to me in the water. Anyway, these things won't hurt you, though. Don't ever interact with a wild animal in a way that it feels threatened, because then it will hurt you, because it's trying to protect itself. That's what happened to the crocodile guy. The Australian guy that used to do all the shows with the animals, and he got hit by a ray in his heart. And he really knew better, and I'm not picking on the guy. He was a lot of fun, but he made the animal feel endangered, and never want to do that, because that's when you're going to get hurt. So I was the one that was against the beach. This guy was towards the water, so he was safe. So I didn't have to worry about him feeling threatened. I was the one that felt threatened. Anyway, see, they can get pretty large. And here's a really good shot of the gill slits that you can see there as the water comes through there. The next one that's on that particular page is the sawfish. And the sawfish has a body like, sort of like a shark, only he's flatter, okay? But he has this long nose with spikes on him, like teeth. And they say that they'll actually, they get over 20 feet long. And then they'll actually go through fish, a school of fish like this, and thrash with their saw and wound the fish, and then go back through and eat the fish. And they can also use that to get crustaceans out of the bottom, is what it says. So they'll dig shellfish out of the bottom. Oh, and the rays all do eat shellfish, not just crustaceans, but also shellfish. Let's see. And these things, it says that there's a population in a landlocked in Lake Niagara, which is interesting. The other thing is these can get 20 feet long and weigh 800 pounds, but they'll be in shallow water. They like warm water, and they will come in and be in shallow water. So I don't think you see that many of them anymore, and I think they're protected now, because you don't see many of them anymore. The next one that particular, oh, this is the underside of the sawfish. And please notice once again, there's the gill slits. You don't see that in fish, but you do see that in rays and skates, that the gill slits are on the underside, and then they have spiracles on the top side to bring in the water. There's spiracles. Let's see if I can find you another shot that shows you the spiracles. Here's the spiracles. See, right in here, there's a place where the water goes in, and then it goes over the gills, and then it comes out through here. Now, if you think about it, this guy isn't on the bottom, he's a swimmer. But this guy could be on the bottom, and some of these other guys could be on the bottom. So if they were pulling water in from the gill slits on the bottom, they'd be pulling in a bunch of dirt, and they'd suffocate. So God designed them so they have these spiracles on the top, which brings the water in the top, and then they can exhale, you know, so that the water goes out of the bottom, and then that's what's pushing out into the sand. So they're not pulling up all that sand into their gills, which is a good thing. These are spotted eagle rays, and they tend to be seen in pairs or groups. I saw them on hens and chickens, which is, that's the right reef. We were on hens and chickens, and the particular day we were there, it was early in the year comparatively, so it was probably in June at latest. And I think they were breeding, because I noticed we saw several pairs of spotted eagle rays go by, and then I saw a pair that their tail was that short. No kidding. They looked like this, only they had a short little stubby tail, so I assumed that they were skates, and they were paired. So it wasn't that one was damaged, it wasn't hurt. And they also were paired and going through. And I don't know if they're also called spotted eagle skates. I tried to look that up and didn't find it, but I've seen these, and they're gorgeous to see. So anyway, and then the last one listed on that page is the electric ray, and this is an electric ray, and apparently he has two kidney. Notice how short he is, and he looks more like a skate to me, doesn't he to you? Because he's got that short little tail, and yet it's called a ray. And I'm not exactly sure why that didn't make skate. Doesn't tell us here. But anyway, wouldn't you have called that a skate from that short little stubby tail? Anyway, But this is a ray, and notice here's his spiracles, once again, for the water to come in. But this guy has two kidney-shaped electric organs located on the pectoral fins, and they are able to give a shock of 200 volts to something like a fish that they're trying to eat, and then they'll go ahead and eat the fish. They tend to, and the book says that all of these rays tend to stay kind of half buried in the sand at night, and then, in the daytime, and then they'll come out to feed at night. So are they diurnal or nocturnal? They're nocturnal, that's good, okay. And so I think that was the last, oh wait a minute, was it? No, one more plate we were supposed to do. If you go over, that's right, to page 85. On page 85. Okay, look over here for a minute for me, please. In biology, if you haven't taken it already, some of you have, I know some of you haven't. In biology, we'll learn there are 3 ways that organisms, animals, can bear young. If they lay an egg, like you and I would think of as a normal fish would lay eggs, or like a bird, like a chicken lays an egg, it's called oviparous. And this parus actually means to bear. So parus, just for those of you that want to know, means to bear, like bearing a baby. And ovi, ovo or ov, equals egg. So when it's oviparous, then it's laying an egg. Okay. If it has the baby live born, it's viviparous. And that viva, that stands for life or live. And so viviparous individuals are live born, and they're specifically live born from a placenta. So I'm going to put live from placenta. Now, what's a placenta? Well, the placenta is actually where the mama's umbilical cord attaches to the baby, and we end up with a belly button after we're born. That's where the placenta fell off. So between, like with mammals, like us and horses and dogs and cats, when we have a baby, the baby is fed by the umbilical cord, which goes from the mama's body to the baby's, what we think of as a belly button, and then it feeds it and takes away the waste products through the umbilical cord between the mama and the baby until the individual is born. Okay, that's viviparous. If it's like a chicken or it lays an egg and the egg hatches, that's oviparous. Now, there is one that's kind of a combination of the two. It's ovoviviparous. And literally, they're just putting the two together. Ovoviviparous means live born from an egg. Live born from an egg. Live born from an egg. Now, what does that mean? That means the egg stays inside the mama until it hatches. And then when it comes out, it is already a baby. Okay? So that's what rattlesnakes do. That's what guppies do. So they actually have an egg inside, and when the egg is ready, then, and it hatches inside mama, and then it comes out. That's actually what seahorses do, isn't it? Okay, so that's ovoviviparous. So these are the 3 types of bearing young that individuals can do. And believe it or not, skates and sharks do all 3, which is just strange. Like, all mammals are viviparous, and all birds are oviparous, but sharks and skates do all 3. Pretty weird, huh? This is telling you about the way they lay an egg. When a skate lays an egg, it looks like this, and it's called the mermaid's purse. Okay, and you've probably found this egg case on the beach at some point in time. The baby rays are developing in that egg, and then when that mermaid's purse deteriorates, it lets the little rays out. This is what the little baby rays look like when they come out of the egg, which is the mermaid's purse. That would be the oviparous individuals. And then they can have babies live born from an egg inside, which is that a strange picture where this poor ray is up on the beach having her babies. Okay, and that's a good sized ray. But anyway, so that would be ovoviviparous, live born from an egg. And then the last one you can actually see the placenta between the mama and the baby, and that would be the viviparous individual. So this one would be ovoviviparous, live born from an egg. This one is attached by the placenta. See it? And so this one is considered a viviparous individual. Okay, I know that's hard. If you've had biology already, it's a review. If you've never had biology, you're going to have to know that part for biology, so you may want to write it down. Anyway, okay, so we got through the information and we got through the stuff on the rays. So what do you say we do the ray observation together, and then you will be able to, let's see, the first part of your homework next week, it says to give your drawings of your ray, the drawings of the skate you observed in class, and identifying the certain parts, which we'll go over together. And then you're going to list 3 facts you learned about skates, which should be pretty easy because we got a lot of facts about skates in all this reading that we did. Okay, so I'm going to set up for our skate observation right now. Okay, so as we do our lab, I want you to notice, once again, we're going to look for the eyes, the spiracle, the snout. The pectoral fins are just these. These are the pelvic fins back here. Oh, so I said they were claspers, but they weren't necessarily. They're just pectoral fins. And then the dorsal fin would be back here, and there can be a second one there. This is obviously, well, this says it's a skate's anatomy, and I don't know about you, but I don't see a whole bunch of difference between that and that. Then if we flip, so this would be the dorsal side. This is the ventral side. I don't know if you remember that, but you need to. So when we look at the ventral side, we should find the mouth, we should find the gill slits, and we'll see what else we find there. Okay? Okay, so that's what we're going to be looking for. I am going to first bring it over to show you right here. Okay? And so here he is. Can you see the whole thing? Back him up a little bit. There he is. Okay, so this would be your dorsal surface. This would be your ventral surface. Here, we see his eyes. So there's an eye, and I'm going to tilt him for you. Can you see? Here's his eye right in here. Okay, right in there. And then, so he's got that on both sides. Then he's got the spherical. There's his spherical. Do you see it? You're going to see spiracles on other elasmobranches also. Okay, and this would be his snout up here. So snout, eyes, spherical. Okay, the spherical right there. Then we go back, and I'm going to move him forward so you can see the rest of him. And here, there is his spine, and I'm going to turn this, hopefully so you can see it. There we go. Can everybody see this spine? Listen. Hear it? That's because it really is serrated along the sides like a steak knife. I'm going to try to get out of the way of the light here. Can you see that? Try to get it so you can see. There it is. And let me see if I can get it so you can see it this way better. There it is. Okay, see that? So that's what you don't want to step on. Shuffle your feet while you're at the beach. And look, this goes all the way up to here. Okay? These would be the pectoral fins. So you see them. There's your pectoral fins, which do the swimming. These back here are your pelvic fins. So we got the, let's see, these do the swimming, like I said, okay? And then towards the back of his body, we have the pelvic fins. Then we have his tail, and believe it or not, this is called his stinger. All right, so let's go ahead now, and let's flip him over. Let's start at his snout. So we'll start up here. There's his snout. There's his mouth. And let's go ahead and open his mouth. See if we can get a view of his teeth. Wow, interesting. His mouth, he's got a piece of tissue that holds it down in the center. Do you all see that right there? Right here. And trying to see if I can see anything else in there. I guess his teeth, let's see if those are his teeth down in there. Yeah, those are his teeth. I can hear them. Okay, so that's his mouth. All right, then we go here, and I want to show you, here's the gill slits. Here's one, here's one, here's one, here's one, and here's one. So 1, 2, 3, 4, 5. So we should have 10 of them all together, right? Here's one. No, maybe there's 4. 1, 2, 3, 4. Is that one there? Oh yeah, that's a tiny gill slit. So there are 5 on each side. There it is. Can you guys see it? I'm opening it up. I don't know if you can see that, but it just barely is opening up. Here, let me show you this way. There it is. See the probe went in? That's the 5th one on that side, and that's the 5th one on this side. It opens up. So anyway, we have 5 gill slits on both sides. We have his mouth. Down here is his anus. So that's where he goes potty. These are his pelvic fins down here. These are the pectoral fins. Okay. Once again, here is his tail. Oh, and he does have a dorsal fin. Okay, let me just turn him back like this for just a minute. Can you see his tail? This is his dorsal fin. See it? So he's got a dorsal fin on his tail, because if you laid that down, that'd be on the dorsal side. Okay? Interesting. Okay. Anyway, so I just wanted to show him to you one more time. And here is our skate that we did our observations on. Okay? Remember, this is his dorsal surface. This is his ventral surface. On his dorsal surface, remember, these are the pectoral fins. These are the pelvic fins. His tail. He has a spine, dorsal fin, believe it or not. There's his eyes. Here's his eyes. Here's his spiracles. And then if you flip him over, you see his mouth. We find the gill slits, the anus, and once again the tail. Okay, and we are going to compare these with the dogfish coming up soon. Okay? But that's our skate observation. All right, you guys. Have a very blessed week, and go ahead and do your best to write up your skate observations, and I'll see you next week. God bless y'all. I miss you. Bye.